All right, good morning, everybody. Why don't we get started? This is an important lecture today because what I'm going to try and do is kind of bring things together. Bring things together from our studies of higher level brain functions, brain areas important for declarative memory, memories for facts and events, as we learned from this man, patient HM. And identification of these key medial temporal lobe structures that are critical for declarative memory. So we talked about these areas. Many people are interested in these kind of high-level mental processes, and we're starting to try and pinpoint these mental processes into brain structures and brain circuits. This was kind of the first big one, medial temporal lobe structures and memory. And then, of course, we moved on to the structures involved in fear conditioning, and here we focused on this structure here, the amygdala. Okay, so that's neuroscience, neurobiology, doing lesions, trying to figure out whether there's selective impairments by specific lesions. But what I hope you are starting to think about is, well, okay, so now we know that medial temporal lobe is important for declarative memory, and amygdala is important for fear conditioning, because if you damage these two, you don't get either declarative memory or you don't get fear conditioning. But what exactly are these brain structures doing? What are the cellular, what are the synaptic underpinnings of this? And for this, we go back to, we talked about not just the hippocampus and the amygdala, but we talked about multiple brain areas that are important for various kinds of plasticity. But what we want to ask is, what exactly is happening here at the synapse that underlies these higher level cognitive functions? So this is really what fascinates many people about neuroscience, and I would venture to guess that that's why many of you came to take this class. We're at the crossroads of neurobiological or mental functions, higher mental functions, and biology. What is the biology underlying declarative memory function? What is the biology underlying fear, our understanding of fear? And for that, we go back to the synapse. So here we're going to go back and use everything that we learned about the basic synaptic functions and ask, well, what could change at this level, at the cellular level, let's say, to enhance a synapse? So fear conditioning is an enhanced response, for example, to an auditory stimulus. What might be happening here that could enhance a synaptic response, if we assume that synaptic responses and cellular responses are underlying this plasticity that we learned about in fear conditioning? So as you remember, we have presynaptic and postsynaptic elements. Can anybody give me some examples of changes that might occur at the presynaptic element that might either enhance synaptic transmission or decrease synaptic transmission? What's happening here? Anything, any scenarios? What can change in the presynaptic terminal to either enhance synaptic transmission or decrease it? Any ideas? Yes. Perfect. Increase or decrease in the amount of calcium coming in this voltage-gated calcium channel. That's perfect, because we're going to come back to exactly that modulation right there. So if you increase the amount of calcium coming in through these voltage-gated calcium channels by, for example, putting more channels in there, what would that do? What would increasing calcium do to the presynaptic terminal? Any ideas? Yes. Very good, yes. A lot more, she said, so when more calcium comes in, that would allow more of these synaptic vesicles to fuse with the presynaptic membrane and let out more neurotransmitter. Why? Because calcium is the secret ingredient here that is causing these vesicles to fuse. And of course, if you have fewer calcium, voltage-gated calcium channels and less calcium comes in, you have less of a signal and less of these synaptic vesicles will fuse. Okay, that's great. Let's hop over the postsynaptic membrane. What do you think? So here, what do we have here? We have receptors for the neurotransmitters. Can you think of ways in which changing something about this postsynaptic membrane would change synaptic transmission. Any ideas? What could you change about the postsynaptic membrane? Yes? Perfect. You can block the receptors. So if you have a receptor antagonist, or you could pull them out of the postsynaptic membrane, that will decrease your response. Conversely, you can shove more receptors in the postsynaptic membrane, and that will give a bigger response on this postsynaptic side. We talked about excitatory postsynaptic receptor potentials and inhibitory postsynaptic potentials. Changing the number or the efficacy of these receptors on this postsynaptic side will change the EPSP or the IPSP depending on what those channels are doing. Okay, so you can see that this is going to be certainly going to be one of the things that we'll go back to, and we will certainly be talking about pre- and postsynaptic membrane mechanisms on the final exam, because we're really trying to pull it together in this lecture. Okay, so those are great examples. Now let's just go over all the possible examples of things that might happen. So first, physiological changes at synapses may store information. I say that because we assume that all of these mental processes, declarative memory, fear conditioning, emotional processing, are all happening because of electrical activity and synaptic activity. So physiological changes at synapses may store this information, this new declarative memory or new fear conditioning that you're learning. The changes could be presynaptic, postsynaptic, or both. Actually, you could imagine that mechanisms can both increase the number of voltage-dependent calcium channels and increase the number of receptors, excitatory receptors on the postsynaptic side, and that would really ramp up the synaptic response at this particular synapse. So it can be presynaptic, postsynaptic, or both. And changes can include increases in neurotransmitter release that we talked about through increases in voltage-gated calcium channels, or effectiveness of receptors. Effectiveness in terms of numbers of receptors, or you could enhance receptors in a variety of other ways. So let's just look at this at a very much more detailed level. So here is your typical synaptic terminal, axon terminal, presynaptic terminal. Dendritic spine is the postsynaptic terminal, and you have this postsynaptic receptive area. This has receptors in it. So just imagine that this little section right here is full of receptors. And with a stimulation of this one axon at the level of the dendritic spine, you get a postsynaptic potential. This is actually an excitatory postsynaptic potential. We're measuring, remember, this is a slide from the previous sections. We're measuring at the postsynaptic potential here, the yellow in millivolts going up. We start at negative 60 millivolts, the resting potential, and excitatory postsynaptic potential brings the membrane potential closer to zero or above zero. And you could also get inhibitory postsynaptic potentials going down here. So again, we're going back to all of the stuff that we learned in the first section of our class. But here we get a small excitatory postsynaptic potential, PSP. This would be millivolts here, and this is time over here. And what we're showing here is just the response from the activation of this one axon terminal. But we're saying here's some things that could happen after learning or experience, after you've formed a declarative memory, after you've done fear conditioning. You could have more neurotransmitter release. That could be one of the changes. So here we have only three little, let's say these red dots are the vesicles released. Here we have 1, 2, 3, 4, 5 vesicles released. You could have bigger or more sensitive postsynaptic membrane. So the presynaptic membrane is the same, but you have more and bigger postsynaptic membrane with more receptors here. Or you could have a whole synapse that enlarges. So the presynaptic terminal gets bigger, there's more synaptic release, and the postsynaptic membrane gets larger, and there's more receptors here. And the result is the same stimulus after the learning experience causes a larger excitatory postsynaptic potential. Okay? So you really need to understand all these different possibilities and understand what it means to change, how these changes would result in an increased excitatory postsynaptic potential. Yes?
Why would a bigger postsynaptic membrane matter if there's more neurotransmitter?
Yeah, that's a good question. Why would a bigger postsynaptic membrane do anything if there's still only 3 packets of neurotransmitter released? That would have to do with the affinity of the receptor. So if there are more receptors there, there's a higher probability that even a smaller amount of neurotransmitter can hook up with the postsynaptic receptor, and that will tend to give you a bigger response. Okay? Any other questions about that? Okay, great. So what other things can change? You could change interneuron modulation. So before training, you might have this interneuron here, but it's not doing very much. And so you have your regular excitatory postsynaptic potential, but something happens after training such that you get activation of this modulation. Is activating the presynaptic terminal, and this modulation actually can change the number of the amount of neurotransmitter released, giving you an increased postsynaptic potential. Various neurotransmitters that can stimulate the presynaptic terminal or can stimulate molecular mechanisms inside the presynaptic terminal can result in increased neurotransmitter release. So it could be internal mechanisms that just increase neurotransmitter release, for example, increases in number of voltage-gated calcium channels, or you could have this modulatory effect where various neurotransmitters can come and affect mechanism inside the presynaptic terminal to then stimulate more neurotransmitter release. So 2 different ways you can get that. Now, in addition to these differences in pre- and postsynaptic activity at the synapse, you could also have larger structural changes. Okay, what do I mean by structural changes? New synapses could actually form as a result of learning. Okay, and or something could be eliminated as a result of learning, and we're going to be talking about particularly elimination, specific examples of neurobiological systems where we can demonstrate this elimination with a certain form of learning. And training or experience might also lead to synaptic reorganization. So what does that look like at the synaptic level? You could form new synapses where there are only 2 synapses to this target area, and now this synapse, this axon develops a little axon sidebar, and you get excitation of both these possible terminals here. So new synapses to this target area are formed. You can get rearrangement of synaptic input, where this axon has inputs to 2 postsynaptic terminals, this only has 1, and then it rearranges so that this one kind of retracts and then this one goes here. So this one, this particular axon will have a larger influence, and this axon has a smaller influence. So that would be an example of rearrangement. Now, using these examples, particularly formation of new synapses, rearrangement and decrease and retraction of synapses, let's just go back and think about how this might work in examples that we've already talked about in neuroplasticity. So how does these examples apply to situations we've already discussed from previous lectures? And let's now go back to our somatosensory plasticity example, where we had this animal with usually 5 digits, 1, 2, 3, 4, 5. The 3rd digit here was removed surgically. And we have what we called an expansion of the cortical representation. So this is a representation now of the part of the cortex that responds to D5. D4 has expanded into what used to be the D3 region. D2 has also expanded into what has been the D3 region, and D1 is right here. So what might be happening here as you see? What might be the cause of this expansion in the examples that we just talked about? What could change as a function to make this happen? Yes?
So T4 and T2 create more synapses between cells and would retract.
They would retract from the D.
If T4 synapses would create those
Yes.
synapse form T2.
So this is going into the D3 region, right? So say that one more time.
So they would make more synapses between T4 and T2.
Okay, so another way to say that is that the D4 synapses would move into the D3 region, and this would become what used to be D3 right here. This used to be D3, now becomes either D4 or D2. So what you have here is something like this. New synapses coming in. This is D3, what used to be D3, but there's no more inputs there. So this all retracts. And what happens is, let's say this is D4, this encroaches into at least part of the region where D3 inputs used to be. Is that clear? Is that clear? Okay. No? Is that a no? Okay. Okay, good. So trying to kind of move into an even deeper understanding of something we talked about, examples of plasticity. So now we not only know that cortical representations change, but we have a more detailed understanding of exactly how they're changing. That we could have encroachment of inputs into areas as other areas retract. So here's D3 retracting, here's D4 coming in. We can say the same thing about D2 coming into different areas of where D3 used to be, D2 coming in, and D3 retracting. Okay, now let's do another example. The other example that we talked about from the somatosensory lecture was overt stimulation of, or excessive stimulation of D2 and D3. And D4, D5, D1, they're just not stimulated very much. But every day the monkey comes in and he does this task where he has to stimulate D2 and D3. You get lots of stimulation there, and that results in expansion of D3. It takes over part of where D4 is and takes over part of what D1 is. Can somebody describe for me what is happening synaptically there? Yes? Yes. Exactly. So it's exactly the same example, same kind of situation as the previous one. You get retraction of the D1 inputs, and the D2 come and innervate this area and expand out into it. And here you get retraction of D4 and expansion of the D3 into this input. Okay? So again, just a more detailed understanding at the synaptic, at the cellular level, what is underlying these forms of plasticity. So this is kind of the crossroads that we're trying to get at. It's really a deeper understanding of not just that medial temporal lobe equals memory, but what are the cellular underpinnings of this change, this kind of mental change or behavioral change that we see. I remember something, or I can't remember anything anymore. What are these areas doing? Okay. So now we need to go back and talk a little bit about the history of how these ideas came to be developed. And really one of the major figures in our understanding of cellular plasticity is a man named Donald Hebb. He wrote a very influential book called The Organization of Behavior, a Neuropsychological Theory. Now, he was really, really smart. this guy had very little or much less understanding of the physiological processes, yet he came up with these basic ideas about what should be happening at the cellular level. These were theoretical ideas. He came from a neuropsychological, or really a psychological point of view. He studied behavior and all the different ways that animals and rodents can learn, and hypothesize about what might be happening in their brain without being a neurophysiologist himself. So what exactly did he say? Hebb's law that he developed, again, just looking at behavior and thinking about logical processes that might happen between neurons. Hebb's law was that synapses grow stronger when the presynaptic neuron repeatedly causes the postsynaptic cell to fire. Okay? So it's not just the presynaptic cell fires and you just get a little EPSP that doesn't fire the postsynaptic neuron. You have to have firing of the pre and the post together. He said that is what would cause change, and that's what would cause enhanced responses together. The maxim that I want you to remember from Hebb's law is that cells that fire together wire together. Okay? Very famous law, easy to remember, very likely to be on an exam. And here is an example. And this is from a chapter we didn't go over explicitly, but this is from figure 7.22, so go back and read that section in the book. This is an example from development, but it could apply to any form of learning, memory, and plasticity that we're going over in these parts, these later parts of the class. So here is the example from development. We have an input. Let's say this is from primary visual cortex cell, and we have inputs from coming from the left eye, stimulated from the left eye. Let's say this is from the LGN, and we have inputs from the right eye. They're both synapsing on this visual cortical cell. But then we go in and we do a trick, a mean trick. We close the right eye. So we put a little patch over the right eye. So the right eye is not stimulated at all. Sorry, the right eye, my right eye. The left eye is open all the time. So what happens is this left eye is getting lots of stimulation. You get lots of concurrent presynaptic excitation that ends up firing, here are the action potentials, the visual cortical cell. And as you get more firing here, the right eye is covered. You get very little firing here. So inputs from the right eye are not stimulating this. The result of this pattern of activity is strengthening of these synapses. And look how strengthening is represented here. You get more presynaptic elements from the same cells go in here. That means that a given stimulation will stimulate the postsynaptic cell even more and result in higher firing from the left eye. And you get retraction of these inputs from the right eye, suggesting that the right eye is not, will not be able to fire this neuron at all. So this idea of more inputs that's causing postsynaptic firing of the cell, resulting in stronger synaptic connections, that is Hebb's law. What he's saying is that you need both presynaptic activity and postsynaptic activity that happen on a regular basis to get this strengthening happening. That was his hypothesis. That was just a hypothesis. Yes? No? Okay. That was just a hypothesis. And again, theoretical. Now we need to search for actual neurobiological examples of this particular law, as well as neurobiological examples of these kinds of theoretical changes in the synaptic organization, the synaptic firing, and the structural organization of the synapse. Okay, where's that going to come from? And that comes from Nobel Prize winning work that Eric Kandel at Columbia University did. He actually did that in collaboration with Tom Carew, who is now a new dean and researcher here, right here in the Silver Building at NYU. Both of them doing really classic work on a very, very simple system, okay? So think about it for a second. This example was in the visual system where you know how complex it is. You have the LGN with different layers representing different eyes, the right eye or the left eye, going up to layer 4 of cortex. These synapses are not, it cannot be easily segregated to stimulate one or the other. And so the insight that Eric Kandel had was to move to a much simpler system, to be able to study very simple forms of learning and memory, but demonstrate them and show them at in a very robust way. And his simple system was the Aplysia. This is a sea slug, okay? It's Aplysia californica, and it's found in California. Was used back in the 70s when Eric Kandel first started this work to today, still used to study the cellular and molecular underpinnings of simple forms of learning and memory. You think, well, what is that useful for? Well, it turns out that these basic principles that he was able to demonstrate very, very clearly in a simple system have now formed the basis and formed hypotheses for being able to study them in complex systems, and we're going to talk about that in the last part of the lecture. Okay, so what are the advantages of this simple slug, the Aplysia? Well, it has many fewer nerve cells. In fact, we could identify all the nerve cells that there are. They are wired up in a much simpler way. And they can create, because we know all the neurons, we can create much more detailed circuit maps for particular behaviors, and there's really little variation between individuals. There are not outgoing aplysia and shy aplysia. They're just all aplysia. Okay, what is studied in aplysia? Habituation is a very simple form of learning that we've all experienced. Habituation is this phenomenon where initially a novel stimulus that elicits response will elicit less and less and less of a response. Okay, so we're going to go over an example of habituation in aplysia. Can anybody think of an example of sensory habituation that they experience? Some stimulus that's new at first, and then you just don't get used to it at all, and you just stop responding to it at all. Not nearly as effective. Yes? 14th Street traffic, okay. So are you saying that early on in your NYU career you were very stimulated by 14th Street traffic and you're not stimulated by that anymore? Oh, hearing. Okay, so perfect. Auditory habituation. So the traffic on 14th Street kept him up early. It was activating his auditory system, not allowing him to sleep. And with time and with lots of sleepless nights, his auditory system habituated, and now that same stimulation is not strong enough to keep him up at night. Perfect example. Any other examples? Habituation. We go through it all the time. Yes? Perfect. When your eyes adjust to brighter light. So first you go out into the bright sunlight and you're squinting and you can barely see, but just after a few minutes your eyes can adjust, and that happens. We talked about pupils get smaller, and the responses to the bright light modulates, and you're able to do that. That is a form of sensory habituation. Fantastic. So this is a form of habituation, simple form of learning. All of this is using the same principle of habituation, and now we can study it in a sea slug that isn't nearly as complex as your auditory system or your visual system in humans. What we're using is sensory habituation to a squirt of water. Aplysia they live in the surf and they get squirted with water all the time. Now we're using, in the lab, we use a water pick to squirt the water on the a part of the aplysia called the siphon, which is like, it uses the siphon to expel waste from its system. And if you squirt water on the siphon, it causes a fear reaction or a safety reaction that is a retraction of its gill, its respiratory system. It's like pulling everything in safety. If something's happening, if something's squirting me, I'm going to kind of hold tight and retract my gill. But after repeated squirts with the water pick on the siphon, the animal retracts the gill less and less. It's like, okay, I know you're not going to hurt me now, so I'm not going to retract my gill anymore. So here is the beautiful aplysia. Here's the head. Here is the siphon, the little blowhole that will excrete waste. And here's the gill underneath. This is underneath the blob of the aplysia. And within this blob, there are also nerve cells that make up its nervous system. And this is a form of plasticity, habituation. We know now that habituation is caused by synaptic changes between the sensory cell in the siphon. So we have sensory cells that are innervating the siphon that can detect, just like sensory cells in our fingers, they can detect movement and water all around the body. It has a somatosensory system just like we do, just much more simplified. So there's a sensory neuron in the siphon, and there are synaptic changes that occur between the sensory neuron and the siphon and the motor neuron that retracts the gill. It turns out that there is a monosynaptic connection between the sensory neuron in the siphon and the motor neuron that retracts the gill. Okay? Very simple system. And we know what's happening there is not just that we see, okay, behaviorally this happens, but the neurophysiological or the cellular change that happens is that there's less neurotransmitter released in the synapse that results in less retraction. Okay? So let's look at the circuit that has been worked out in the aplysia. You have your water pick stimulation, the squirt of water on the siphon. The siphon has axons from a sensory neuron. Its cell body is right here. And those neurons go, those sensory neuron axons go and synapse with the motor neuron that then has acetylcholine receptors that does gill withdrawal reflex. Okay? And so what we see is that here's the normal circuit. Nothing bad's happened to the aplysia, but now we water pick it with the siphon. And there's a big retraction because there's lots of neurotransmitter release here and the gill withdrawals. You do that 10 times in a day, and at the end of the day, the gill is like just withdrawing just a tiny bit. It's like, don't even bother me. And we can go in and look at exactly what's happening here. And what we see is actually what they did is physiological studies at these neurons, very detailed intracellular studies. And what they found was there was a change and a decrease in the sensitivity of the calcium, of the voltage-gated calcium channels. Voltage-gated calcium channels in aplysia neurons, just like in the neurons we talked about in the beginning of class, are mediating the release of neurotransmitter. So what happens in this kind of form of what we call short-term habituation, habituation that happens over the course of a couple of hours, is that you get a desensitization, less activation of the voltage-gated calcium channel after this repeated stimulation, and that just results in less neurotransmitter being released for the same exact stimulus. Okay? So this sensory neuron is being activated just the same by all the water pick responses, but because the voltage-gated calcium channels are less active here, there's less calcium getting in for each stimulation, and therefore there's less neurotransmitter. Okay? So for the short-term habituation, you get a modulation of the voltage-gated calcium channels. That is a physiological presynaptic change that happens that's underlying habituation. Now they noticed another thing that happens with habituation. If you bring the little aplysia back for 4 days in the lab and give him 10 habituation squirts each day, what happens on each successive day, he habituates faster and faster and faster. That is, those responses go down earlier and earlier and earlier. And by the end of those 4 days of 10 stimulations a day, the animal is barely responding to this water pick at all. Okay, so and that decrease of response can last for several weeks. Okay, so what happens? Is it that you get a decrease in the calcium, voltage-gated calcium channels for several weeks, or there's something else happening when you have a long-term habituation? And so that's exactly what I just said. And here is what happens. With long-term habituation in these aplasia synapses, you don't get a modulatory effect at the presynaptic terminal. Instead, you get actually a structural change in the sensory motor neuron, to sensory presynaptic neuron to motor postsynaptic neuron connection. What does that look like? Instead of having these 3 connections from your sensory neuron onto the motor neuron, you get retraction, and there's many fewer synapses from the sensory neuron onto the motor neuron. Okay, so that is the long-term change. There are structural changes. So you say, okay, well, that's great for aplasia, but I still want to understand how declarative memory works. How is this change in neurotransmitter and this change in structure going to help me there? Oh, I should also mention that, again, this is the first stage of the basis of the Nobel Prize that Eric Kandel won. This is a very, very simple example that I'm giving you, some of the earliest work that they did. But it was seminal. Why? What was the seminal? I want you to understand why he would be given a Nobel Prize for this. And it's because that all of these ideas of synaptic changes associated with a behavioral change were mainly theoretical before he started to understand, he started to work these out. So he, by using the very, very clever idea of moving to a much simpler system, still had a clear behavior he could monitor, habituation, long-term habituation. So he had a behavior, and again, he was doing this cool thing in bringing the behavior and understanding it and studying it at a cellular level. So cellularly, looking at the changes, you could show physiologically that the calcium-gated, voltage-gated calcium channel was changing in the short-term habituated animals. And for long-term habituated animals, he can show structurally that before long-term habituation, you have these beautiful, extensive axonal terminals onto the motor neuron. And then with long-term habituation, you get just this dinky little projection. He was able to show this with Golgi studies showing the structural change. Again, this was really, for the first time, bringing the structural, behavior to the level of a cellular understanding. And we won't have time to this, but the other reason why he won the Nobel Prize is he didn't only look at the cellular functions, the anatomy and the kind of cellular voltage-gated functions, but he was able to go deeper and look at the molecular changes that are happening both presynaptically and postsynaptically and identify some of these molecular cascades that are also changing long-term. We'll get into a little bit of that at the end, but I just want you to appreciate that this came at a critical time. This was seminal new information, and it was really an acknowledgement of what a amazing idea it was to bring all of these ideas to a simpler system and to be able to show cellular things at a simpler level. And of course, all of these principles that he was able to show in this simple system can now be shown are now being studied as hypotheses in more complex systems. Yes?
So, in this specific example, there was nothing postsynaptic that changed. It was all pre.
Yes, these examples are presynaptic changes. There are postsynaptic changes that I'm not going over, but these examples are all presynaptic. But in the simple aplasia, as in the more complex systems that we'll talk about, the hippocampal slice system, changes are typically include both presynaptic and postsynaptic. But the simple example that they were, some of their first examples were mainly presynaptic. Okay, so So we want to go from this simple aplysia that won a Nobel Prize. Great, lots of new ideas and great pathways identified there. But what we really want to understand is what's happening in my hippocampus, in my entorhinal cortex, when I'm learning something new. As you guys are learning all this information about cellular underpinnings of learning and memory. This is an episodic memory that you will be tested on at the end of the class. What is happening in your hippocampus that is allowing you to take this in, to understand, and hopefully to be able to recall it perfectly at our final. Okay, and there is a very important phenomenon that was discovered in the 1970s that gives us a clue as to some of the cellular properties that may be working at the level of the hippocampus. This phenomenon was discovered in actually the rabbit hippocampus as they were doing stimulation and recording experiments in the hippocampus. And the phenomenon is known as long-term potentiation. Long-term potentiation, or LTP, is defined as a stable and enduring increase in the effectiveness of a synapse, okay? So we talked about habituation, that is a decrease of synaptic efficacy. Here we're talking about LTP, which is a long-lasting increase in synaptic efficacy. And the secret was they were looking at responses to a brief stimulation, so they would stimulate axons within the hippocampus and record in cell body layers and record the response. But then they gave a special kind of stimulus called a tetanus. This is a brief increase in electrical stimulation. It's actually multiple bursts of activation. So you can give a little burst, single, 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 and just look at the response. And the responses to these single, short stimulations will not change. A tetanus is like high frequency stimulation multiple times, like 100 hertz, stop, 100 hertz, stop, 100 hertz, stop. It is multiple high frequency stimulation. And it tends to trigger thousands of action potentials. When they use this pattern of stimulation, they didn't do it all the time. They did it for a relatively short period. They gave a tetanus, this a short acting high frequency stimulation. And then they went back to giving the individual stimulations. And those responses to the individual stimulations then increased, and they stayed up for a long time. That increase in response is called long-term potentiation. And first I'm going to show you where exactly they did these recordings and stimulations in the hippocampus, and then I'll show you what it looks like on a graph. Okay, so we're talking about recordings in the hippocampus. Here is your rodent. This could be a sheep brain as well. The C-shaped hippocampus here. We're taking a horizontal cut and we're looking at a slice of hippocampus that looks just like this. Now here, when we talked about the parts of the hippocampus that were abnormally kind of lined up in schizophrenia, if you remember for our last lecture. I told you about the different subfields of the hippocampus. So now I'm actually going to ask you to learn these different subfields. Here are different cell layers. It's kind of a 2C configuration, one C here and another C here. These yellow, orange areas and green areas are filled with cell bodies. And so this part of the C, the middle part of the C, is called the dentate gyrus. Okay? The dentate gyrus, the next structure moving along the next C, is called area CA3 and area CA1 here. You don't have to learn CA2. CA2 is very, very small. So we have dentate gyrus here, cell bodies of the dentate gyrus. Then we move on to the cell bodies of area CA3 and area CA1. CA are subfields of the hippocampus. It stands for cornus ammonis, which refers to horns of a ram, because people thought that these looked like kind of ram's horns. And what you need to know is that the cell bodies within the dentate gyrus are called dentate gyrus cells, And their axons are called mossy fibers because the axons look like moss. They're so intricate in the axon terminals. These dentate gyrocells terminate via the mossy fibers on area CA3. CA3 cells then send their axons through axonal systems that are called the Schaefer collaterals. Anatomists like to name different brain structures after themselves, so Schaefer named these. And CA3 terminates on the dendrites of the CA1 pyramidal cells. Okay? So we have mossy fibers here and Schaefer collaterals here. Mossy fibers come from the dentate gyrocells, and Schaefer collaterals are the axons of the area CA3 cells. So what we're doing in long-term potentiation is typically we're stimulating the mossy fibers, so you can put your stimulating electrode in a particular region right in here and know that you're getting lots of mossy fibers and give a nice big stimulation here. You get lots of activity, lots of stimulation to the mossy fibers, and then we're putting a recording electrode here in the CA3 area, and we're measuring the postsynaptic potential, the excitatory postsynaptic potential, not just of one synapse, but of the full population. This is called the population excitatory postsynaptic potential, or EPSP, population excitatory postsynaptic potential. And so you can imagine with a strong stimulation in those mossy fibers, you'll get a huge EPSP, or population EPSP. And what we're going to be doing here is measuring the slope of this response. This is a response in millivolts over time, and we can measure the slope of the response. So if you have a really high slope, then that means it's going up really fast. If you have a low slope, then it's not a very strong reaction. So that's what we're going to do here. Now here, what we're looking at is the slope of the EPSP in millivolts per millisecond, and low slope down here. This is a slope in response to just a single stimulation. So single stimulation in the mossy fibers, right here, stimulating here, single stimulation, recording here, looking at the population EPSP in CA3. This is population EPSP because, as we'll talk about in a second, this is an excitatory synapse using glutamate as its neurotransmitter. Then, right here, we give that tetanus response, a high frequency stimulation response, and then we go, this is just happening at minute 30. This is literally, it lasts for 10 seconds. And then after the tetanus, we start giving the same single stimulation. And you can see the slope increases. So that means that that population response has increased substantially, and that slope is maintained for, look, 30 to 120 minutes. For 90 minutes after this very brief stimulation, you get an increase in the synaptic response. And the people that discovered this said, this is cool. We know, by the 70s, we certainly know the hippocampus is important. We know about HM. We don't know how it's doing it, but this really striking plasticity could be underlying some of all of the learning and memory functions that the hippocampus is doing. Okay? Okay, so now what I want to try and do is relate this change in the slope of the EPSP to something, a theoretical idea that we talked about a little bit earlier in class. And that is Hebb's law. The idea that synapses grow stronger when the presynaptic neuron repeatedly causes the postsynaptic cell to fire. And in fact, Hebb's law, which was developed way back in the 50s, 40s, and 50s, was exactly what these researchers were thinking about when they started to think about the implications of this finding. Why? Because this high-frequency tetanus stimulation is stimulating a lot of presynaptic terminals at the same time that is likely activating lots of postsynaptic terminals. That brief presynaptic, postsynaptic co-activation then results in this higher stimulus response. This seemed to be an exact example of what Hebb's law says. They were seeing stronger synaptic responses when lots of presynaptic, when they stimulated lots of presynaptic neurons at the same time that they were causing postsynaptic cells to fire. Why would that be? What are the cellular underpinnings of this? Why was this happening? Now we know it happens. Why does it happen? And now we move into an understanding at the cellular level that might make this change happen, okay? So to go from just early response to a strengthening of response, because this left eye is being stimulated a lot, and the left eye stimulation is activating the visual cortical cell a lot. Okay, so why is this the case? This is the case because there's a particular special kind of postsynaptic receptor in the postsynaptic membrane. So we talked about presynaptic mechanisms. Now this is all about the postsynaptic membrane, a very special kind of receptor. But before I go into that, I have to just remind you that we are now talking about, let me just go over this slide. I basically said all of this. The people that discovered LTP in the hippocampus. This was actually discovered in the rabbit hippocampus, but now has been studied extensively in the rodent and the mouse hippocampus. These cells exhibit this form of plasticity very, very easily. Sorry, one more thing I forgot to say, that you can see LTP by stimulating the Mossy fibers and recording in area CA3, or by stimulating the Schaefer collaterals and recording in CA1. Multiple synapses throughout the hippocampus exhibit very, very easily long-term potentiation. Okay. So synapses in LTP behave like Hebbian synapses because tetanus drives repeated firing. Postsynaptic cells, target cells, fire repeatedly due to the stimulation. You can record these postsynaptic cells and not just record their EPSPs, but show that they're actually spiking as a result of the tetanus. And synapses grow stronger than before. All these characteristics that define a Hebbian synapse. Okay, I just said this. It occurs at several sites. And now I want to remind you of glutamate as a neurotransmitter. So we talked about, this is going back to our lecture on neurotransmission and drugs. And we talked about all these different kinds of neurotransmitters, the more modulatory neurotransmitters, and the critical excitatory and inhibitory neurotransmitters in the brain were glutamate as the excitatory neurotransmitter and GABA as the inhibitory neurotransmitter, if you remember that. We want to focus on glutamate right now. Again, it's the major excitatory neurotransmitter of the brain. It is the workhorse of your brain. Anytime a cell, the majority of the time a cell is getting excited in your brain, you're using glutamate. And we know that there are at least two critical types of glutamate receptors. Remember, we talked about different kinds of dopamine receptors, different kinds of acetylcholine receptors. There are two key kinds of glutamate receptors that will be play a big role in our understanding of LTP. One uses AMPA, and the other uses NMDA. There's actually three: AMPA, kainate, and NMDA. We're going to talk about two major kinds of receptors: the AMPA receptors and the NMDA receptors. Those are the two main glutamatergic receptors I want you to be familiar with, and it'll be clear from the next few slides. Okay. It turns out that the CA1 region has both NMDA and AMPA receptors. What happens is glutamate that's coming from the Schaefer collaterals this is actually, this can be CA1 and CA3 have both NMDA and AMPA receptors. So what I'm going to be telling you about the CA3 to CA1 synapse is also true for the dentate gyrus to CA3 synapse. Okay, so CA1 region has both NMDA and AMPA receptors. Glutamate first activates AMPA receptors, but NMDA receptors don't respond until something special happens. So let's just look at the picture first. So here is our axon terminal. Let's say this is an axon from CA3 to CA1. This is a Schaefer collateral axon. Works just like all the other axons we talked about. Voltage-gated calcium is activated when an action potential comes down and releases neurotransmission. Now, in normal neurotransmission, most of the receptors that are activated are AMPA receptors. AMPA receptors let sodium in. Sodium will depolarize the cell. Okay, so again, going back to all of our synaptic mechanism stuff, you really do need to understand this to understand the full explanation of LTP. So in normal synaptic transmission, glutamate is activating the AMPA receptor, and the NMDA receptor is still here on the postsynaptic side, but under normal conditions, NMDA is not active. Why? Because it has a blockade within this receptor of magnesium. Okay? So magnesium keeps it blocked, usually, during normal synaptic transmission, and you get activation, sodium coming in. The thing that will get the NMDA receptor going, and this is the special thing to get LTP. To get LTP, you need NMDA receptor activation. That means you need somehow to get this magnesium blockade out. Mg2+ refers to magnesium. How do you do that? You do that by getting so much activation in the postsynaptic membrane by the regular AMPA receptors that you increase the depolarization of the postsynaptic terminal so much that the magnesium block is relieved. So if this postsynaptic terminal depolarizes to a certain level, then magnesium will pop out of here and the NMDA receptor will become active. How would you do that? You would do that by getting a whole bunch of glutamate out here to really activate all possible AMPA receptors. You could also do that by not only activating this little dendrite, but all the dendrites on this axon that are receiving inputs from CA3. So you can imagine that when you put a stimulating electrode and titanically stimulate with high frequency the Schaefer collaterals, you're going to be getting a huge depolarization of lots of this postsynaptic membrane. First, because AMPA receptors, all possible AMPA receptors are being activated, and then that gets up the postsynaptic membrane potential so high because sodium is coming in that all the magnesium pops out of the NMDA receptors. Okay, so now we have magnesium out. It's unblocked of the NMDA receptors. What's so great about that? Once magnesium is out of the NMDA receptors, what happens is you get calcium that comes in through the NMDA receptors. Okay? Calcium will also help depolarize the postsynaptic membrane, but more than that, calcium works to do something special. Calcium will actually activate different second messenger pathways. We talked about second messengers in the metabotropic receptors. Calcium is a very, very important signal here, and it is going to activate a second messenger system called the CAMK2 system. You just need to know that name. It's a second messenger molecular cascade system within the postsynaptic membrane of the cell. This is now we're in area CA1. Big deal. Now we have CAM-K2 activated. What does that do? Well, that does something very special. There are internal stores of AMPA receptors. These are the green regular AMPA receptors in this postsynaptic membrane. What activation of CAM-K2 does is it then sends a signal to these AMPA receptors to insert into the postsynaptic membrane. So once you get more activation of the postsynaptic membrane such that the potential increases, you get calcium coming in. Calcium activates CAM-K2, a second messenger system, that will then tell more AMPA receptors to insert into the postsynaptic membrane. And what is that going to do? What does more AMPA receptors do? What will that result in, do you think? I now have more AMPA receptors in the postsynaptic membrane. What's gonna happen? Yes? More glutamate can bind with the postsynaptic AMPA receptors, and so we have more glutamate binding. What happens then? More activity. Why? Because activation of the AMPA receptors lets in more sodium. Okay, so if you have just more of these AMPA receptors, even the same amount of glutamate will cause a higher deactivation, depolarization of the neuron, and that hopefully makes it clear why you get this long-term thing. This is a structural change. I now have significantly more AMPA receptors in my postsynaptic membrane. They're not changing, they're not going away. They've been inserted now, and so now with the same stimulation, I'm getting more of a postsynaptic activation, okay? Okay, so these are just the explanations. Let's just make sure that they all make sense to you. NMDA receptors at rest have a magnesium block on their calcium channels. After partial depolarization, the block is removed. Partial depolarization happens when the AMPA receptors are activated, letting sodium in and depolarizing the postsynaptic potential. This removes it allows the magnesium block of NMDA receptors to be removed and allows calcium to enter in response to glutamate. And so here we have, does it show? Okay, here we have more AMPA receptors in the same postsynaptic membrane, causing a change in activation. So now we have, even in mammals, even in this complex structure like the hippocampus, we have a cellular model for how neural responses can change and how they can change pretty long term. This is a structural change, at least in the number of receptors in the presynaptic terminal. Now, are we able in the hippocampus to show, just like we did in aplesia, retraction of particular axons or addition of axonal components? And the answer is no. Why? Because the hippocampus is still, in the mammal, is still too complex to be able to see. It's like looking for a needle in the haystack. But this model of AMPA and NMDA provides a cellular basis for Hebbian plasticity in the mammalian hippocampus, and that has been for a long time our model for starting to understand how the hippocampus can change in response to new learning. Okay? Now, I haven't told you at all how to relate this LTP to your learning, what an NMDA receptor does. So you all just learned what an NMDA receptor does, and you'll be able to answer hopefully some questions about that. That is new learning. That is dependent on your hippocampus. Can I tell you exactly how LTP, are you all going through LTP right now? Still unclear. This is still a model. but it gives you a flavor for the kinds of approaches that we're trying to use to understand the cellular basis of learning and memory. Let's see. Okay. Okay, any questions? This is a complex set of responses, but I need you, and I will be testing you definitely on LTP and how AMPA and LTP contribute to Hebbian plasticity. You're going to have to tell me, probably in an essay, how that is the case. Anybody have any questions right now? How that relates? Why NMDA allows you to have Hebbian-like plasticity? Can anybody tell me why is the NMDA receptor so important for Hebbian plasticity? Why does it explain it? Yes?
It allows more AMPA receptors to activate and therefore characterize the
Right. It allows more AMPA receptors to be activated. More generally, and this is a very important point, how do we relate the NMDA and AMPA story to Hebbian, the Hebb's law? That you have to have both presynaptic and postsynaptic cells firing at the same time. Well, you have to have presynaptic and postsynaptic cells firing at the same time because you need not only presynaptic activation, but you need enough activation of the postsynaptic cell to get the magnesium block. So that implicates that this is highly active. There are lots of activations here, probably causing spiking. So this the features of this channel, that it has a magnesium block until there's enough activation in the postsynaptic terminal, actually is a perfect explanation for Hebb's law. You need pre and postsynaptic activation concurrently because you need to get enough momentum, electrical momentum going in the postsynaptic neuron to pop that magnesium out. So actually, when they discovered the NMDA receptor, it was a really elegant confirmation of the theory that Hebb came up with before he had any idea of AMPA receptors and NMDA receptors. He just had this theory. But this receptor and the features of the receptor, that it's not active until there's this postsynaptic activation, explains perfectly Hebb's law. Does everybody get that? That Hebb's law requires both pre and postsynaptic activation, and that's exactly what you need to get activation of NMDA. That's why the NMDA receptor is a perfect kind of explanation for Hebb's law. Okay, think about that. Ask me later if you want to, if you need to. Very, very important concept, and we will be talking about modulation of memory on Monday.